everyone. This is our third virtual session in our Grant Writers Toolkit series from the RCMB ECP Working Group. This session is entitled Demystifying Study Section for Independent Awards. We have a great session lined up for you today. We're going to start off with a didactic from our NIH program officer colleagues who have come here willingly and with lots of information for you. Dr. Yan Hu is going to introduce them shortly here. We'll follow that session with a question and answer session for you to all get out any particular questions you have with regard to grant review for independent awards and the changes that will be upcoming in 2025. We then will proceed with our mock study section. We did send out a grant, the specific aims and scientific approach of an ESI, early stage investigator grant. And if you didn't receive that, I will put it into the chat again for you to review afterwards. But for the purposes of the mock study section, you're not required to have looked over it. And then we'll have a short Q&A to end the session. I just want to thank our faculty participants from the RCMB, Dr. Chen from the Cedars-Sinai Medical Center, Dr. Shoan Sengupta from Children's Hospital of Philadelphia, Dr. Rachel Knipe from Harvard University Massachusetts General Hospital, and Dr. Josh Englert from Ohio State University. Of course, I'm going to thank our ECP working group members, Dr. Yan Hu, Dr. Maria Basil, and Dr. Jermaine Katzen, who have helped work on programming this particular session. And then certainly Tiana Lena, who has helped us throughout managing all of these sessions and facilitating our administrative needs through the ATS. And with that, Dr. Hu, you can go ahead and get started for the first portion of this session.
Okay, thank you. Thank you, Scott. So we are very honored to be able to invite both Dr. Marisol Espinosa-Pintucci and Dr. Roya Kalantari from the NIH NHLBI. So both are program officers, and then they will give a didactic presentation about the details of the review process, scoring system, and especially important parts for the ESI status applicants. And also there will be some newly updated criteria for the coming year. And then welcome both doctors, and then the stage is yours.
Great. Thank you very much. My name is Roya Kalantari, as been introduced. I'm one of the program officers in the Division of Lung Diseases.
Hi, everyone. I'm Marisol Espinosa-Pintucci, and I'm also Program Officer for Training and Career Development in the Division of Lung Diseases. And so I'm going to start off today with the first half of this presentation, and we will be talking about navigating the NIH grant submission and review process. Next slide. So our talk today will cover the following topics: the anatomy of the review process, what happens to a grant when it is submitted, how reviewers are chosen for particular grants, the NIH scoring system and recent changes, how and when investigators might interface with a scientific review officer or a program officer, and important points for early stage investigators. Next slide. So what happens when a grant is submitted to NIH? It generally takes approximately 9 to 10 months from submission of an application to award of an NIH grant. Once the application is submitted, it will go through several steps, including referral and review, pre-award and award process, post-award and monitoring, and then eventually reporting. Before you submit a grant, you need to make sure that that you review the Notice of Funding Opportunity, NOFO, completely, because it contains very important information about the purpose and the background and description of the program, and also contains the application review information. Next slide. So going back to the question of what happens once your application is received by NIH, including how we determine which study section it will go to for peer review and how we assign it for funding consideration. So all grant applications submitted to NIH go to the Division of Receipt and Referral, DRR, within the Center for Scientific Review, CSR. After receiving the applications, DRR checks the applications for completeness, determines the area of research, and assigns it to a specific NIH institute or center. DRR also assigns applications to specific study sections, also known as scientific review groups or review committees, that have the specific expertise to evaluate the scientific and technical merit of an application. The NIH uses a dual peer review system, or 2 levels of peer review, which is mandated by law in accordance with Section 492 of the Public Health Service Act and federal regulations. The NIH policy is intended to promote a review process that strives to be fair, equitable, timely, and free of bias. The first level of review is carried out by the scientific review group, also referred to as a study section, composed primarily of non-federal scientists who have expertise in relevant scientific disciplines and current research areas. The Center for Scientific Review coordinates the reviews for most R01s, fellowships, and small business applications, as well as some program announcement, PAs, PARs, and requests for applications, RFAs. Institute and centers coordinate the review for applications that have institute-specific features such as program project grants, training grants, career development awards, as well as other RFAs. The second level of review is performed by the Institute Center and Center National Advisory Council. Councils are composed of both scientific and public representatives chosen for their expertise, interest, or activity in matters related to health and disease. Only applications that are recommended for approval by both the Scientific Review Group and the Advisory Council may be recommended for funding. And final funding decisions are made by the NIH Institute or the center director. Next slide. As mentioned, initial peer review meetings are administered by either the Center for Scientific Review or one of the NIH ICs with funding authority as specified in the funding opportunity or the NOFO. Each NOFO specifies the review criteria and considerations that will be used in the evaluation of applications submitted for that NOFO. And each scientific review group is led by a scientific review officer, an SRO, and the SRO is the NIH extramural staff scientist and designated federal official responsible for ensuring that each application receives an objective and fair initial peer review, and that all applicable laws, regulations, and policies are followed. The scientific review group is composed of a chair who serves as a moderator for the discussion of the applications under review, peer reviewers, and other NIH staff. An initial peer review evaluates applications for the scientific and technical merit of the work proposed, overall impact, which we will discuss in a little bit, and appropriate justification for human subject protections, inclusion across the lifespan, and vertebrate animals. Next slide. So how are reviewers chosen for particular grants? The NIH peer review system's continued excellence depends on the ability to recruit and retain the most accomplished, broad thinking, and creative scientists and experts to serve as peer reviewers. Peer reviewers, and I speak broadly in terms of both ad hoc and members of study sections, are mostly non-federal extramural scientists. They are active scientists and recognized authorities in their field. Expertise is a major consideration when developing a roster for a study section, and each scientific area reviewed needs expert representation. There also needs to be balanced representation of reviewers in a study section. There must be diversity with respect to geographic distribution, gender, race, and ethnicity, and this includes recruiting reviewers from different institutions. And then there's fairness. Candidates must be dedicated to high quality, fair reviews. Fairness and objectivity are the most important criteria for a reviewer. Conflicts of interest are also managed. For example, the reviewer may not be in the study section if the reviewer's name is on the application in a major professional role, is a member of the NIH Council, or the reviewer or a close family member would receive direct financial benefit if the application is funded. After identifying potential reviewers, other information is used to determine eligibility, such as extramural funding, history, publication history, and prior review experience. For each application, there are 3 or more reviewers to provide an in-depth assessment, written critiques, and criterion scores. And very important to the review process, assignments and conflicts of interest must remain confidential. Next slide. The review criteria and the NIH scoring system. So each notice of funding opportunity will provide information about the application review information and review criteria. And this will be located in section 5 of the NOFO. The application review information and scored review criteria will differ depending on the grant mechanism. And there are 3 general categories for all applications, and this will not change with the simplified peer review framework that will be implemented for the RPGs or research project grants that Roya will discuss next. Applications receive an overall impact score determined by review criteria, and reviewers provide an overall impact score to reflect their assessment of the likelihood for the project to exert a sustained, powerful influence on the research fields involved. The first category of scored review criteria for RPGs, this includes the current 5 criteria, require criterion scores on a scale of 1 to 9. And these scores play a role in determining the overall impact score. There is also the additional review criteria, such as human subjects, and this does not require criterion scores, but does influence the overall impact score. And then there are the additional review considerations, such as budget and period of support, and they don't require scores, and they also don't have an effect on the overall impact score. Next slide. So evaluating the overall impact, reviewers consider the different criteria as well as the additional review criteria mentioned. The NIH grant application scoring system uses a 9-point rating scale, 1 equaling exceptional and 9 to equal poor. In whole numbers, there are no decimals for criterion scores and final impact scores for all applications. NIH expects that scores of 1 or 9 will be used less frequently than other scores, and 5 is a good medium impact application and considered an average score. During the review, after the review of the application, all members provide final scores privately, and their overall impact score for each discussed application will be determined by calculating the mean score from the eligible members' impact scores and multiplying the average by 10. The overall impact score will be reported on the summary statement. So I'm going to hand it over to Roya, and she will discuss the simplified peer review framework for most of the research project grants, RPGs. Next.
Yeah, so yeah. Thanks, Marisol. So before I get into what the changes to the review framework are for research project grants, I kind of want to cover quickly why this is happening. So really the overarching goal of this is that NIH wanted to ensure that peer reviewers can better focus on answering the key questions of should and can the research proposed be conducted, and to really mitigate the effects of reputation of the PI and the institution by contextualizing the review to more clearly be within the framework of the research being proposed, rather than having somebody's reputation kind of carry it forward, even if there are flaws. And also to kind of reduce reviewer burden a little bit by moving some of the additional review criteria, so those components that are not scored, that are really more about policy compliance, and shifting that onto NIH staff to review instead. So I mainly want to note that this is for applications submitted for due dates on or after January 25th. So if you're planning on submitting prior to January 25th for a due date prior to that, this does not apply to your application, but it will from January 25th onward. There's also a webinar that just got posted about yesterday that will be taking place on April 17th about all these changes. I highly encourage you to register to attend that, because they'll go much more in depth about these changes than I will. So what are the changes? So as Marisol mentioned, there's 5 current criterion score components to an application. These are significance, investigator, innovation, approach, and environment. And so that's what the current grouping is. But now what's going to happen is those 5 are getting shifted into 3 factors. And so these 3 factors, factor 1 is importance of the research, factor 2 is rigor and feasibility, and factor 3 is expertise and resources. So factor 1 is now going to be a combined of significance and innovation. Factor 2 covers approach as well as some additional review criteria that I'll get into on the next slide. And factor 3 is investigator and environment. And so what you'll see is that factors 1 and 2 are going to be scored as has been previously done, given criterion scores between 1 to 9 with the same metrics used as previously. However, factor 3, covering environment and investigator, will not be given a criterion score. And instead, this will be a drop-down menu where the reviewer will either say the expertise and resources are appropriate or not appropriate. When not appropriate, they can then give some comments as to why they think it's not appropriate. But this is again trying to pull away from having reputation carry a grant in ways that it shouldn't really do that. And so the way that they think about this is really, factor 1 is should the work be done, 2 is can it be done, and 3 are there appropriate resources. For the additional review criteria and considerations, so the top of this slide are the ones that affect the overall score but are not scored individually, and the bottom is those that are discussed after scoring and don't affect scores. You can see that these are shrinking down a little bit. So for the review criteria, study timeline and inclusion of women, minorities, and children for human subjects in clinical trials, those are actually being moved into factor 2. They'll still have to be addressed, but they're going to be included into that criterion score as well. For additional review considerations, you can see that shrinking down significantly as well. So reviewers will only now have to comment on authentication of key biological or chemical resources and budget and period of support. Foreign organizations, select agents, and resource sharing plans will all be moving to be fully reviewed by NIH staff. So that is the changes that are coming down the line. So the next topic that we were asked to cover is kind of when to talk to who at NIH about what. So this is just a really quick overview of who the main players are that you'll interact with at NIH. So there's program officers, scientific review officers, and grants management officers and specialists. So program officers like Marisol and myself, we're there to really help you pre-application and post-application and post-award. So pre-application, you can write to us to talk about scientific alignment of your study or other questions you have about, whether or not it's the appropriate funding mechanism that you're applying to, responsiveness to review criteria. After review, once you get your summary statement, where the assigned program officer on your application is the person you can go to talk about resubmitting, any questions you have about the summary statement, or any additional material you need to submit should it look like your application will be funded. And then after award, to discuss any changes. So, we're the folks reading your progress reports. So that's the time when, we may also be reaching out to you with questions and things like that. Scientific review officers, so you already heard a little bit from Marisol about the role of SROs and managing study sections. So really your interaction with them is a little bit more narrow as an applicant, where the only time you'd really interact with the SRO is once your application is submitted and prior to review. So if you're, you have additional documentation you want to submit, post-submission materials, or if you have some new concerns about conflicts of interest with anyone on the panel that you want to raise with the SRO, that would be who you'd want to reach out to. And then finally, the grants management officers and specialists. These are the folks that are in charge of budgetary policy. So POs are the scientific policy, grant specialists or the budgetary policy. So they're really the ones that you want to talk to either before you submit or after award about any questions you have about, you know, allowable budgetary or where, you know, if you want to use funds in a certain way on a type of grant mechanism, whether or not you're allowed to use those funds in that mechanism in that way. And once it's been awarded, if you need to move some funds around, if that's allowable, if you have, you know, carryover or a lot of funds left over. And also importantly, grants management specialists are the arbiters of the grant folder that holds all of your official documentation for your active grant. So it's always a good idea to include both the program officer and grants management specialists on any communications you have with us about your currently active grant, because they're the ones that are going to be in charge of officially documenting everything. And so the last topic that we're going to cover is on early stage investigators and what this is and why it matters. So just briefly, the early stage investigator status. This definition is very specific for NIH. It means an investigator who has completed their terminal degree or medical residency within the past 10 years and has not been awarded a substantial competing NIH research grant, meaning like an R01, that you were the PI on the competing application, right? So if that hasn't happened yet and you're within 10 years of your training, then you should be an ESI. This status is calculated in ERA Commons based off of the information you put in about your training past. So if it doesn't look correct in your ERA Commons profile, make sure that you go back in there and look at what you've put in as far as your training history, because it automatically calculates it that way. An important thing though is that you can request extensions to your ESI status for various life events. We provide a 1-year extension for childbirth. We also gave lots of extensions for COVID shutdowns that happened, but also, you know, family care responsibilities or anything else that takes you away from the bench for an extended period of time. There's an NIH-wide committee that reviews these requests, and so you can put in a formal application for extension, and they'll review that, and then case-by-case basis will grant extensions as needed. So, but why do you care about extending your ESI status? So there's kind of two aspects of why ESI matters. One is at the peer review level. You know, so peer reviewers are thinking about ESI applications a little bit differently, right? So an established PI with a long track record, with tons of publications, you know, we don't want ESIs to be held up to that standard because that's just not possible. You just haven't been in the field as long, right? So peer reviewers are looking more at potential academic and research backgrounds, you know, weighing things a little bit differently there. And also where feasible in the larger study sections, SROs will try to separate out all the ESI applications into their own bunch so that, you know, kind of can help the reviewers gear shift to think about the group of applicants that they're reviewing. At the funding level, NIH, you know, we designated this ESI status because we wanted to attempt to create greater parity in success rates among early stage and established investigators, because we were noticing that success rates for ESIs was not as high. And so some of the ways that we do this is, for example, here at NHLBI, we tend to give a 10 percentile bump to ESIs. So, you know, say if our pay line was at the 14th percentile, ESIs get 24th percentile. And even for ESI, an ESI second application, so you're not actually called an ESI anymore once you get that R01, but your second application or renewal of your R01, if it's within a couple of years of that first R01 where you were an ESI, we give a 5 percentile bump. So we kind of track that to help bridge that process into being an established PI. And finally, the last early career program that I want to bring up, this isn't really necessarily ESI related, but it's sort of in the same window and time frame of your career, is the early career reviewer program. So this is an opportunity to participate as a reviewer on a study section. As an early career scientist, you need to have at least 1 year of full-time faculty at the assistant professor equivalent, you know, have some senior authored publications and have not held an R01 or equivalent, so a major research grant yet, but you must have submitted a grant proposal. So, you know, an R21, a K, anything like that would qualify, or even if you submitted an R01 and it wasn't reviewed favorably, that could still count. It's about the process of submitting the application that matters more. And so this is an opportunity for you to really participate. I've never heard a bad thing about doing this. It gives everybody insight as to what a study section actually looks like. And so with that, we are done. Hopefully we didn't go too far over, and we are happy to take any questions.
Thank you for both of your detailed explanation. I do get one question from the chat box from Dr. Sunat. Would you please explain how exactly the percentile is determined? Is it specific to the study session, or is it specific to the institute, regardless of study session?
Do you want to tackle that one or me?
I can try to tackle it. So let me just, where is the question? It is, I think it is specific to the study section. And so, but I'm not exactly sure. The idea is that it's
So if I could jump in, it kind of depends on how long a study section has been around. They'll use different baselines for calculating percentiles. So like if it's a standing study section with a long track record, they may use the history of funding or history of application scoring from that study section to baseline the percentiling calculation. Other study sections that are maybe ad hoc, they could use other baselines. It might be the entirety of CSR's scoring history and use that as a baseline for percentiling calculations. I hope that makes sense.
If I may add something on top of that, Roya, how I understand it is that for the standing study sections, they take the last 3 cycles, all the scores from that to figure out what the percentile. So it's a kind of a rolling every 3 cycle. For the SEPs, they do the same thing for a group of SEPs because the numbers aren't as much. But I think for the ad hocs, I don't actually know how they do those.
Yeah, it depends because with an ad hoc study section, they just don't have that historical data to be able to baseline it.
Yeah.
Okay. Thank you. So there is 2 other questions, both are about ESI status extension. One is asking whether how to use the COVID mechanism to extend their ESI status, since most of us already experienced the COVID period during our training. Pretty much everyone have this gap. So basically the question is, should that be applied for now or towards the end of your ESI status period? I hope I explained the question well.
So I know like a couple years ago, the ESI Extension Review Committee was asking people to kind of wait until the end of their ESI period to put in, or closer to the end, right? Not so that it lapses, but closer to the end to put in the ESI extension requests if it looked like they needed it, just because, you know, otherwise they were getting inundated with 1 million requests and it might not even be needed. That person could maybe apply for an R1 the next year and get it and be fine when they have 5 years left or something like that. So I don't know now how they're considering COVID extensions and what that looks like. It honestly might not hurt to reach out to that, you know, put in an application to that committee and see what they say. It's not a big application process, but you need to provide sort of dates of impact, severity of impact, how much time was lost, and that kind of information. And then they assess it that way.
Okay. So another question, probably this investigator is trying to apply to another institute. So she's asking whether the percentile bump for the renewal is only provided in NHLBI or by the other institute.
Each IC handles that differently. So you would need to look at each IC's policies on funding to determine what that is. You know, some institutes will even give percentile increases to what are called new investigators, which is a broader category than what ESI is, and HLBI doesn't do that. So, you know, it just depends. You need to check at their web pages and see what they have.
So with that, I would like to ask a question for myself. So I wonder, for the new criteria, would that change the structure of the grant in the future? Do we still put in the different sections in the same way, or in the same order?
Yes, so everything is staying the same on the applicant end. I'm really glad I went to a session on this morning because they confirmed that there. Everything is staying the same on the applicant end. There is no changes to the way that you write your application, but you know, the way the review criteria are listed will be different. So thinking about how those review criteria are structured, and you know, this will be in the new funding announcements as they're released. Look over that section and kind of see how everything's been rephrased and see if that makes you think about the way you write your application differently. But from an administrative perspective of how the application is structured, that's not changing.
Okay. So if there's no more questioning, I'd like to thank both Dr. Kalantari and Dr. Espinosa-Pintucci for your time and detailed explanation. It's really helpful and help us to orient the next step. And please feel free to drop if you are busy. So next, we're going to jump to the second part of our session. So I'm going to pass on to Scott.
Thank you. And thank you to our program officers. You're welcome to stick around, but you guys don't have to as far as the session is concerned. Thank you very much for your time. So we'll move on next to the mock study section portion. I'm going to go ahead and throw the grant back into the chat. So for those of you who weren't able to get it, you can download it now. Again, you don't have to have reviewed it to get benefit from this portion of the session. I am going to preface with the fact that this particular grant was produced by a very excellent investigator at an institution with some very significant resources. And the particular grant in and of itself, while an early stage investigator, an ESI first submission, is really truly exceptional from that perspective. And I think the expectation for most first submissions from an early stage investigator would not necessarily be this, I guess, level of mechanism and preliminary data, and as well as resources. So don't be distraught if you read through this and feel maybe overwhelmed. Even some of our very successful colleagues on the mock study section here have iterated that this particular grant is exceptional in ways that probably many of us will fail to achieve. So with that being said, we're going to move to this session. Dr. Chen will be serving as the chair of the study section. Dr. Naik will be reviewer 1, Dr. Englert will be reviewer 2, and Dr. Sengupta will be reviewer 3. For the purposes of this session, some of the comments will be accurate, and some of them will be maybe somewhat some free license taken with the grant and the material for the purposes of education. So with that, Dr. Chen, I'll throw it over to you, and I'll be silent.
All right, thanks, Scott. All right, so we are going to review mechanisms of regulatory T cell mediated recovery from severe influenza A viral infections. And I think all the conflicts, we don't have any conflicts for this grant. So the reviewers 1, 2, 3 are Drs. Naik, Englert, and Sengupta. Initial scores, please.
I gave it a 1.
I gave it a 4.
I gave it a 2.
All right, Dr. Naik.
Okay, so this is a new application from an early stage investigator seeking to examine the mechanisms underlying the resolution of lung injury following severe influenza A viral infection, and specifically looking at the role of regulatory T cells in this process. The proposed studies will test the hypothesis that Tregs require the epigenetic regulator protein ERF1 to maintain their methylation status, which is central to their identity and their pro-repair function, and also amphoregulin to induce epithelial repair during recovery from severe influenza A viral infection. There are 3 aims in this proposal. Aim 1 will determine whether ERF1 is necessary to maintain Treg cell transcriptional programs and pro-repair functions during recovery from influenza. The studies will utilize several tools, including inducible Treg-specific ERF1 knockout mice, single-cell RNA sequencing, DNA methylation assays, and gain-of-function adoptive transfer experiments. Importantly, they use physiologic readouts in these experiments of lung injury and repair. Aim 2 will ascertain the necessity of Treg-generated amphoregulin in promoting repair during recovery from influenza A-induced lung injury. These experiments will use additional tools, including Treg-specific amphoregulin knockout mice, and again, gain-of-function through adoptive transfer. There's a nice built-in discovery sub-aim in aim 2 to explore the time window over which Treg-mediated recovery occurs through the adoptive transfer of Tregs at various time points. And then finally, aim 3 will bring in a human component to determine whether transcriptional and epigenetic signatures in alveolar Tregs are associated with 30-day mortality in patients with severe viral pneumonia. These samples will be collected as part of a larger program, gathering additional longitudinal data in well-phenotyped pneumonia patients. So I kind of tried to group them by these new categories. So factor 1, significance and innovation. So this proposal is highly significant. There's high morbidity and mortality with ARDS, especially viral infection, increased ICU usage, and understanding mechanisms here to promote repair and resolution will develop urgently needed therapies. If successful, is highly likely to generate really novel insights into how Tregs promote recovery. There's really compelling preliminary data to support the project, and the PI has nicely shown that the mouse model system that they use is relevant in human disease by showing similar transcriptional and epigenetic changes in response to flu infection in both humans and mice. There's also, in terms of innovation, significant conceptual innovation in the hypothesis that Tregs require ERF1 and amphoregulin for epithelial repair. The observation that mouse and human Tregs show similar changes, as I mentioned, is novel. A role for amphoregulin has been shown in the acute phase of flu, but not in the resolution and repair phase. So that's also novel. Several of the experimental tools are novel, including the DNA methylation assays, the ERF1 transgenic mice, and a novel flow cytometry-based assay to quantify KRT5 positive epithelial cells as a readout of repair. Factor 2 gets to the approach. So I thought there was a really comprehensive and rigorous approach presented. The approach is clear, logical, and very realistic. The grant is really well written, very easy to follow. There were nice figures showing the experimental design for each sub-aim. The preliminary data strongly supports the underlying hypothesis, demonstrated high feasibility and likelihood of success in all 3 of the aims. They nicely added the gain-of-function rescue experiments, their knockout experiments, and I thought they did a really nice job of presenting potential limitations or problems and suggesting alternative approaches or solutions. Their statistical methods were appropriate and the experimental group sizes were all appropriately justified. Overall, I really thought this was an excellent grant, and I had a hard time finding any flaws or deficiencies, which really speaks to why I gave it a 1. And then finally, in terms of factor 3, the investigator is a highly qualified early stage investigator with an impressive track record of publications and early level funding, who's compiled a really outstanding investigative team of collaborators. They are all highly qualified to perform all of the experiments proposed. And finally, the environment is really excellent. And in addition to having all the resources to perform the experiments, this project is part of a larger program of human data, which I thought added a particular strength to the proposal. So that kind of summarizes why I gave it a 1.
All right, Josh.
All right. Yeah, I agree with reviewer 1 that this is a highly significant proposal addressing an important clinical need. from an exemplary EPI with a strong, very strong track record of published data. I also agree that the approach was really clearly presented. It was mechanistic and quite elegant with, paired gain and loss of function experiments. But although the approach was quite strong, I did think there were some weaknesses or areas for improvement. And specifically, I think there were a few missed opportunities to look at the other half of the lung barrier, right? This is focused mostly on how Tregs interact with the epithelium. And I thought there was a potential missed opportunity to look at the endothelium, and it was largely ignored in the proposed studies. I thought this was important because some recent data in the field demonstrates that the lung endothelium can actually regulate Treg function in other models of acute lung injury. Another potential limitation was that, clinically, when patients are admitted with influenza and ARDS, they receive antiviral therapy, and how these drugs may affect Treg identity and function really can't be determined from the proposed experiment, since a group of control mice treated with antivirals wasn't included. I also thought it was worth noting that the NOD2 receptor is known to regulate Treg recruitment and function, specifically inflammation. And I thought, although there were a lot of physiologically relevant outcomes presented, the assessments of inflammation that were described in the in vivo experiments were really quite limited and superficial. And then finally, in the proposed clinical study name 3, I thought one potential confounding issue that was not addressed was the fact that, these patients that recover, i.e. the controls, it's possible that one reason they recover is because they have a more immunosuppressive, highly functioning Tregs at baseline. And I didn't think that was going to be able to be sussed out in the proposed experiments. That being said, on balance, I thought these weaknesses were relatively minor in the context of the entire proposal, and again, agree with about the comments about the investigator and the environment. And so for overall, I thought the likely impact of the proposal was high, and my enthusiasm was also high.
All right, Shan, you got anything else to add?
No, I think I noticed the strengths and weaknesses that have been highlighted by Josh and Rachel. I think that even though I noticed the same weaknesses that Josh has highlighted, I just I weighted them differently. I thought that while some of the endothelial Treg interactions were not highlighted, as an ESI, the investigator is starting out with a broader and clearer mechanistic platform and will build upon it. And therefore, even though I noticed it, I did not, I was convinced enough with the strengths, and on the balance, I thought it was an outstanding application, and I gave it a 2.
Right. We'll open this up for discussion, and I think I'll start things off. It seems that, Josh, I think we all kind of agree on a lot of the strengths, but Josh, you've listed a lot of weaknesses. You stated it was kind of relatively minor, but still your score did not quite reflect that. Can you or the other reviewers kind of weigh in on kind of how important these weaknesses are that you've listed?
Yeah, I think, it's a fair point. I think after hearing reviewers 1 and 3 and, thinking about the balance of strengths and weaknesses, I think my score may have been a little bit low, and I think I will likely, increase it in response to kind of hearing what they said and how they, balance some of those strengths and weaknesses.
I think Josh brought up some really good points. And I just wanted to mention that I think the point about the endothelial component is a really interesting one, but I think it's hard for an investigator to put all the cell-cell interactions into one grant. And I think if they had, for example, also tried to look at endothelial interactions and or other cell interactions, I might have read as too much, and I think this PI did a really nice job of focusing focusing on one hypothesis and designing all the experiments around testing that hypothesis. And it may be that, you know, an endothelial project may be like a separate proposal. So I worry that would have been too much. I do think your point about the antiviral is a really good one, and that would have been a nice arm to include. But I do think it's relatively minor in the overall picture.
Yeah, I want to echo what Rachel mentioned, and I do think that some of the other things that came up, whether the way the injury is being classified, whether it's too superficial, I just feel that from an ESI investigator, having to put together a grant that walks the fine balance of ambition and feasibility, I felt that this was doing a really great job achieving both. All of those aims are, like, all of those ideas are solid, but I can see that if those were all included, I would probably have said that this seems a little unfocused and overtly ambitious for someone's first R01. So while I see those as great points, I'm sure some reviewer somewhere in a manuscript will bring them out and it will get addressed. So I'm reluctant to penalize this otherwise outstanding application and investigator for those things. That's why I think writing about it so that he or she gets the feedback, but still gets a good outcome from this. That's how I at least weighed it.
Great. I think we've had a great discussion. So why don't we move on to additional review criteria, human subjects? Exemption
Included,
4?
fine. Yeah, no problems.
Okay. All right. Inclusion in women, minority individuals across lifespan. It's okay.
No concerns.
No
All right.
concerns.
Vertebrate animals,
Included.
biohazards.
Included.
All right. And this is not a resubmission or revision. So
Nope.
I will try to summarize. So this is an application from an ESI physician scientist who proposes the hypothesis that Tregs require UHRF1 to maintain their pro-repair function and amphiregulin to induce epithelial repair during recovery from severe influenza A infection. So there are a lot of strengths. I think there's a lot of consensus on the strengths. A highly significant proposal on studying the Tregs and the recovery after influenza. It's very innovative studies, and the hypothesis is fully supported by the preliminary data. The approach is rigorous, comprehensive, and includes powerful gain of function and loss of function tools with many physiological endpoints. The PI is outstanding, very impressive track record of publications and funding, and the environment is just built for the success of this proposal. There are several weaknesses, including missed opportunity to study the endothelial compartment, as well as including an antiviral arm in the studies for the murine studies. There were some more detailed analysis of inflammation would have been helpful, and then also the control arm for the AIM-3 human studies might have been a confounder because they're just not quite appropriately matched. But overall, there was a lot of consensus for the strengths of this application. There was divergent comments on the weaknesses, but overall, it sounds like people were feeling the weaknesses are pretty minor, and this application was high impact. Does that sum it up appropriately?
Yes.
All right, good. Final scores, please.
I'll go to a 2.
I'll come up to a 3.
I'll stay at 2.
All right, 2, 3, 2. So your range is 2 to 3. Anyone going to vote outside the range? I'm going to say it's a no.
No.
All right, and then we usually go through non-scoring issues, budget, resource sharing, authentication, select agents for, and justification. That's it. We're done. Great job.
Thank you very much. Thank you, guys. That's so wonderful. Thank you. If there's any questions, you can go ahead and throw them in the chat, or certainly raise your hand or unmute yourself now. I will start with a question, and it does actually go away from the meat of the application to these kind of unscored sections, which we breezed over briefly and really aren't discussed very in any real detail in the process of the study section. But I guess what I'm going to ask is, what are the things that can happen in those sections that can really really tank a grant? Is that a portion of the grant that really could take a really well-performing grant, but they do something in those sections really poorly that leads to its, I guess, a change in its impact or a potential change in funding.
So, the only thing I would say is, for the non-scoring sections, budget. So none of that's supposed to weigh into the reviews. But as an ESI, don't put a $4.99 budget in there. It's probably not the best thing to do. When I was coming through, I was always told do a modular budget as an ESI. But with inflation, that's modular budgets just don't pay enough anymore. And so as long as you fully justify your budget, that's great. But don't try to say, I'm going to do all the single cell and I need to pay all this money out for a $4.99 budget, because that's going to raise some red flags and people's hackles will go up kind of subconsciously. I would just warn you against that.
Peter, I have heard this. I did put it in as a non-modular budget, and I think towards not in the first year, but in the later years, it did go high. It was well justified because of the kind of work we do. The animal numbers are high. But I do think that in study sections, I've also noticed people not just looking at the bottom line, but also looking at equipment and travel, which for some reason in some random applications can be weirdly inflated, which also I think makes, like, again, to Peter's point, it is not as if that'll tank your entire application on the dint of that one point, but it definitely leaves a bad aftertaste. I don't know. That was the sense I got.
Yeah, but the thing is most of the time the non-scoring, they're supposed to be non-scoring, and usually they don't drive anything. And so again, most ESIs aren't going to be putting in these crazy budgets either. Just make sure you fully justify it, and you'll be fine. I've seen lots of ESIs get, the numbers are just creeping higher and higher just with inflation. And so we're seeing much higher budgets now, and most people don't blink an eye. Well, I would highlight a few things when I was reading this application. This is an abnormal application, I'm going to just say, for an ESI. It's abnormal for pretty much anyone that puts in an R1. But this was a very well-written grant. It was so easy to read. And I would like to highlight just a few of the things that this person did. This group is actually very well integrated, and they actually have all their people read their grants. And so I would highly encourage ESIs to always have other people that have had lots of R1s read your grants, because it will give you insights, because you have to remember, you're going to have 3 reviewers reading your grants, and they all come from different viewpoints. And so you can kind of really mitigate a lot of that by having more people read your grant ahead of time. If you look at the figure 2, the schematic, actually, I'm sorry, it's figure 1. The schematic that in the split frames, very simple, very, very clean and simple. And that's how you want to do it. You don't want to have all these arrows going everywhere, because that just means you have a complicated grant that you're never going to solve. And you're going to just get picked apart. It's death by 1,000 cuts. And so, you can see how, they have aim 1 as just one arrow, aim 2 as another arrow, aim 3 as another arrow. That's what you want to do. Just make it nice and clean and simple. You don't want to make it too complicated for yourself, because everything you do in aim 1 has to answer that question. And so if what you're doing is not answering the question, then it's going to hurt you, if anything, because it's just descriptive or whatever. And this investigator did a really fantastic job of getting the tools needed to answer these questions. Had gain of function, loss of function. And then for their aim 3, they have this entire database with biorepository that they dipped into to answer those questions. And so I think you can learn from that. You're not going to replicate this, but you can learn a lot from how this grant was written. One of the other key things I would say is this grant writer is very seasoned in that they actually stated, okay, there's a gap in the literature. The whole amphiregulin Treg, Alex Rudensky published that in Cell several years ago, right? And so he's like, yeah, it was published, but I'm going to look at recovery from injury. I'm not going to look at the acute injury. And what did he do? He got a letter from Alex Rudensky. So that is, what you need to do is kind of attack the issues head on, where the weaknesses are, and go with it. And by failing to mention something about Tregs and endothelial cells, one line, yeah, I know it happens. it's future direction, it's too much for this grant, would have counteracted that reviewer 2 completely, right? And so just kind of, you have to be kind of cognizant and kind of figure that out and just attack your weaknesses and just throw it out there. And so there's kind of a lot of the things I picked up on. The other thing they did really well in their approach, they kept on putting these schematics in, you know, like figure 8 and then figure 11. A picture's worth 1,000 words. I didn't even have to read the grant. I just had to go through those figures and I could figure out what they wanted to do. So it just made it very easy to digest.
I totally agree. I loved the figures. Like as I was reading, I was like, what day are they calling recovery? And it's super clear in all of their diagrams that day 10 is where they're starting all their treatments. And so it's very easy to like look up the details and figure out what they're doing. And I also agree with your point about the Rudensky stuff. They didn't hide from the fact that there's stuff in the literature. They were very clear about what's been done and where they were going to fit into it. So I totally agree.
I would echo Peter's sentiment about, like, you have to really try to put yourself in the mind of your reviewer. And remember, they're reading like 10 of these, and it's not always under like easy circumstances, right? Maybe they're traveling, or maybe it's like late at night after they've spent all day rounding, or who knows. But the easier you make their job, right? They have to fill out these forms and then like complete these reviews. If they can just like take pieces, if you very clearly say, this is why, these are the 3 innovative aspects, and they can then use that to fill out the form. Just, I think that's one of the things that this proposal does really well. It makes it easy. Even like little things, it's kind of like, you know, they're putting in like references to like biohead, like additional review criteria are like woven in there like perfectly. Again, making it easy. And, you know, the one thing I heard someone once told me was, your reviewer should be able to like, your Ames page in particular, should be able to like read through it without having to stop and think about it at all. And like, you can even do that, like you can read it out loud to yourself or to other people. And if you can get through it and it makes sense without stopping, that's how you know you have like a nice, cogent and cohesive Ames page. Because every time, not all grants are like this, and every time you have to stop or like go look up a bunch of references, or like there's like a triple negative in the sentence and you're trying to figure out like it makes it harder for you. And then that's when you start getting, those go in the weakness column of the review.
Yeah, I always tell everyone, just consider reviewers stupid, and you got to force feed everything to them. The more you make them think, the angrier they get, the lower your score goes.
I also like this thing about this grant, which I think has, there's a lot of attention to detail, which is very easy to miss unless you're looking closely, but it's very helpful when you're reading it as a reviewer. You'll notice that all the figures which talk about single cell or show a volcano plot or some other sequencing data, whatever they want to point, they have a very directed way of pointing out the things that they want you to look at. So for example, in figure 13, all the genes of interest are already marked out in a different color, and they are linked with a pathway, so that you don't have to do the work as a reviewer of looking through, is this what they are trying to show? Is this what? So it just makes it very, very easy. So it's a very directed tour through their thing. And the other part, which is also easy to miss, but once you've seen it, you cannot unsee, is that in many of the preliminary data, they not only tell you what they are showing, but they also mention pertinent details, not a lot, but pertinent details of the kit they used and what they did, which for a ESI is very important, because nobody will probably look at it if you've done this a million times and printed 500 papers on this. But when you're an ESI, the feasibility of it, everyone has 12 pages. You don't have an extra page because you're an ESI, but you have to prove that you can do all of these things and you know about them well enough. And so those are really nice, very well done, like, very finely done work where they have proven feasibility without losing real estate on the pages, but also showing that I know this, I've got this, I've done this, and this is the exact detail. Just 3 words or 4 words of what method they used is enough to show, and they've done this repeatedly over many of the preliminary data figures. So that also, I think, is very helpful to prove feasibility as an ESI.
I think, Tanya, I think your question was answered earlier, so partially. So partially. So I guess Tanya had asked for the experimental design diagrams throughout all the sub-aims to were those helpful and impactful? I think we had said that many of them were. Are they worth the repetition?
It depends, you know, it's not a one size fit all. I think this experiment there is very animal based. And so having those diagrams for each kind of experimental design was very helpful. You know, but you don't have to do it for everything. I'm
going to ask, so we technically are over by a little bit, but I think this is maybe an important point. But Rachel, Schoen, and Josh, I don't know if you participated in the early career reviewer program, or if you did, can you comment on that? Also, if you didn't, can you comment about how you became involved with doing a study section and as a, I guess, first kind of study section, how that went for you and how others can do that as well?
I did early career review. It's invaluable. I mean, to be in the room and hear these discussions, you get a reduced number. Like I had 3 proposals, only 1 was discussed. So that made it less intimidating because I wasn't going to have to talk as much. I was reviewer 3. Yeah, I can't say enough good things about it. And, you know, they're happy to have you there generally. Like you're like introduced as the early career reviewer. So people like understand. There's like a link, I don't know if it was put in, but there's a way to apply online and you can apply to specific study sections. I put it in a few years ago and then didn't hear back, and then knew it was getting time for I was getting close to my first R1 submission and I wanted to kind of do that. And so I actually reached out to a couple SROs for the study sections I was interested in, reminded them that, hey, like I applied for early career review a few years ago. If there's ever any way I can be of service, please don't hesitate to reach out. And then, you know, within a cycle or 2, I got an invitation. So
I had a very similar experience to Josh. I had heard about the ECR from a colleague who had applied for an R01 like maybe 2 years or so ahead of me, and she had very good things to say about it. So I applied, but then there was crickets. So I reached out and then I did an ECR, and I also, for circadian expertise, served on a special emphasis panel of some sort during COVID, but the ECR experience was on Zoom, so it was definitely very helpful. But since then, I've participated in person study sections too, and I found them to be really useful experiences. Not as an you can serve as an ECR only once, if I understand right. Later, I was a ad hoc study section member, but I did find both the ECR as well as that being on the study section as an ad hoc member particularly useful and clarifying in many levels that there is no amount of advice that you can get from people that gives you that level of clarity that comes from being in the room. I'm like, oh, this is what so many people have been trying to tell me, and now I get it. So those of who are eligible, I would go home and apply today and then see what happens.
Yeah, I did not do the ECR program, but I was on a in-person like foundation study section for 2 years before I had done anything at the NIH, and that was really helpful to sort of ease in and similarly have like a little bit of practice and exposure to how the discussions went. And then I was asked to be on a special emphasis panel for like a non-NHLBI section, which I only had 1 grant. And so I did a lot of listening and watching. And so I think that helped also just to see how the interactions went and how people presented stuff and how the discussions went. So by the time I was in my first NIH study section for the NHLBI, it had some experience, but it was still a little overwhelming.
Hey, thank you very much. So it looks like Dr. Espinosa-Pintucci put in some information with regards to how to apply for becoming a study section member outside of the ECR program. Otherwise, with that, we're a little bit over, so I am going to call it time. I want to thank our program officers from the NIH for coming and presenting to us, our faculty members from the RCMB Assembly and our ECP working group. Thank you to all of you for showing up and sticking around. We probably had more than 50% of people stick around to the end, which is a good indicator that people were finding value in the session. Our next session is at the end of April, on April 30th. That'll be on proving feasibility and a bunch of additional sections in independent awards. Thank you.